Hello, my name is Dr. Cary Harding. I am professor of molecular medical genetics and pediatrics at Oregon Health and Science University in Portland, Oregon. And it's my great honor today to present to you about our work on cell and gene therapy in phenylketonuria or PKU. I thank you very much for the kind invitation to present our work today. I am also a physician scientist, so in addition to my research, I also diagnose and care for patients with rare metabolic diseases. This is a patient of mine, Victor, who we filmed some years ago. Were I meeting him for the first time for a diagnostic evaluation, I would describe him to have a severe global cognitive disability, microcephaly, and short stature. He exhibits curious repetitive movements of his hands and constant rocking of his upper body. I learned from his medical history that he has a seizure disorder. He also exhibits hyperreflexia and a wide-based unsteady gait. He has a somewhat musty body odor about himself. If I collected a blood sample on him and analyzed the plasma amino acid profile, I would find an extremely elevated concentration of the essential amino acid phenylalanine and a slightly low plasma tyrosine. This pattern is highly suggestive of the disease phenylalanine hydroxylase, or PAH, deficiency. PAH is an enzyme predominantly expressed in liver that converts phenylalanine to tyrosine. Biallelic mutations in the PAH gene lead to PAH deficiency. When that happens, phenylalanine accumulates in the body and tyrosine decreases. Phenylalanine at high concentrations spontaneously deaminates to phenylpyruvate and other phenylketones. The discovery of this disease back in the 1930s was based upon the finding of phenylpyruvate in urine. The presence of this and other phenylketones led to the colloquial naming of this disease as phenylketonuria, or PKU. It took a few years to prove the association with elevated blood phenylalanine, and almost another 20 years until the deficiency of PAH enzyme in liver was proven. There are now other known enzymatic causes for phenylketonuria, so it's most appropriate to refer to this specific disorder as PAH deficiency. Now, almost 90 years after the discovery of PAH deficiency, we still do not completely understand the pathophysiology of the disease. Although the enzyme is mostly active in liver, PAH deficiency does not adversely affect liver function in any way. However, untreated PAH deficiency through the toxicity of elevated phenylalanine has profound effects upon the brain through multiple proposed mechanisms. Fortunately, we don't see many individuals like Victor anymore because since the early 1960s, all infants in the US and most of the medically advanced world have been screened for elevated blood phenylalanine after birth and if found to have PAH deficiency are treated with dietary therapy. Contemporary therapy for PAH deficiency involves restricting the dietary intake of phenylalanine through restricting the amount of intact protein consumed. This means no meat, no dairy products, no normal grains, breads, or pastas. Most fruits and vegetables are okay. All the nutrients and energy eliminated from the diet must be replaced, and that is accomplished by providing a phenylalanine-free medical beverage, essentially an infant formula that has all the fat, carbohydrate, vitamins, minerals, and amino acids other than phenylalanine. Diet therapy prevents the major manifestations of PAH deficiency, but this synthetic diet is expensive, unpalatable, and must be maintained throughout life. Even with dietary therapy, the incidence of attention deficit and neuropsychiatric conditions are higher than in the general population. Off-diet, affected individuals risk the development of an as yet incompletely understood white matter disease. The fetus of a woman with elevated blood phenylalanine is at extremely high risk for birth defects. And for these reasons, my PKU patients really would like to have a permanent cure. Any therapy that lowers blood and brain phenylalanine can positively affect the outcome in PAH deficiency. There are several different treatment approaches that are either clinically available now or are in various stages of investigation. In my lab, we've chosen to work on two approaches that permanently restore PAH activity in liver. First, therapeutic liver repopulation with hepatocytes, also known as cell therapy, and second, gene therapy. In the lab, we test out these methods in a mouse model of PAH deficiency. Simple transplantation of wild-type hepatocytes into PAH-deficient mouse liver yields less than 1% repopulation and no decrease in blood phenylalanine. We demonstrated in this paper that the key to making this work was to give the donor cells a selective growth advantage that allows them to grow and expand after transplant. Here we used a genetic trick that I don't have time to fully describe, but under these selective conditions, following transplantation of PAH-positive hepatocytes into PAH-deficient mice, the serum phenylalanine concentrations plummet once the expanding percentage of PAH-positive cells reaches a therapeutic threshold. That threshold is at about 10% repopulation. The blood phenylalanine levels drop to the normal range, here depicted in gray, once the liver had been repopulated with at least 10% PAH-positive hepatocytes. This is good news for both cell and gene therapy because it means that restoration of liver PAH activity in as few as 10% of hepatocytes will restore blood phenylalanine levels to normal. There is an open clinical trial of human hepatocyte transplantation for PAH deficiency at the University of Pittsburgh, and to date, 2 adults have undergone this treatment. My friend Dr. Ira Fox provided me with these slides from the operating room, where he successfully placed a catheter through the recanalized umbilical vein from the umbilicus into the portal circulation, where he has injected radio-opaque dye to illuminate the liver circulation. He introduced isolated human hepatocytes through that catheter into the liver. Prior to the transplant, in order to give the donor cells a growth advantage, the liver had been preconditioned with focal gamma irradiation. On a later liver biopsy, anti-PAH immunohistology revealed a few isolated PAH-positive cells. This was associated with a small decrease in the blood phenylalanine concentrations. Further work to improve the engraftment and proliferation of the donor hepatocytes is ongoing. Now turning to gene therapy. Gene therapy is simply using genetic material, most commonly DNA, as a drug to treat a disease. So how does this work? The most straightforward form of gene therapy is called gene addition. In this approach, we don't actually alter the patient's PAH gene. We just add in a normal copy of the PAH gene to the hepatocyte. There are a variety of ways to do this, but the treatment that has been done most successfully takes advantage of nature's best gene therapists, which are viruses. The natural function of a virus is to infect a cell, and they carry their own DNA into the host cell. The virus we work with is a specific flavor of a virus called adeno-associated virus, and we'll abbreviate it AAV8. We have engineered this virus to carry a normal PAH gene, along with a liver-specific promoter and other sequences that are necessary to express the PAH gene. We can produce the virus, which are the small particles in this picture, inject these into the animals or humans with PKU, and they will infect the liver to make PAH. After a single IV injection of our engineered virus, their liver begins to produce PAH, and their blood phenylalanine levels immediately plummet into the normal range. High blood phenylalanine inhibits the production of melanin, so PAH deficient mice are tan colored rather than the expected color of black for their genetic background. Once their blood Phe decreases, their coat color turns back to black. So after a gene therapy experiment, we don't even really need to do the blood Phe measurements. We can tell who was cured by the color of the fur. There are now 2 separate AAV-mediated gene addition trials in adults with PAH deficiency, one by Homology Medicines and the other by BioMarin. They each use 2 different flavors of AAV that differ by the capsid proteins coating the surface of the viral particles, but they both are designed to target the liver. Both trials are phase 1/2 dose escalation studies designed primarily to evaluate safety of the treatment with efficacy as a secondary outcome. So far, there have been no major safety issues. 2 of 6 individuals treated in the homology trial have responded to the treatment. No efficacy data have been released yet by BioMarin. With this gene addition approach, the piece of viral DNA in the hepatocyte does not associate in any way with the genetic material of the cell itself. If something causes the hepatocyte to divide, the cell's genetic material is copied and carried through to the two daughter cells, but there's no mechanism to preserve the gene therapy vector DNA and it will be lost. Hepatocytes in adults gradually turn over and are replaced. We do not yet know how long AAV gene therapy in adults will last. Furthermore, this approach won't work in young children because in the growing liver, hepatocytes are dividing very rapidly and they quickly kick the viral DNA out. We know this from many animal experiments, including in PKU mice, in which we lose the gene therapy within a day or so if we inject it into newborn mice. So how can we design a more permanent gene therapy? What if we could find a way to go in and physically fix one or both of the mutations on the patient's PAH genes and restore their own PAH production? This is called gene correction, and that's the focus of our lab work currently. The data I will highlight here was generated as part of the PhD thesis work for Dr. Dalen Richards, who's now a medical student at WSU and who has been involved in organizing this event today. This picture is from the most recent meeting of the National PKU Alliance, the major patient advocacy group for PKU who funded Dalen's project. One of the hottest technologies in the field now is gene editing using CRISPR-Cas9. You've likely heard about this technology. It was first applied to editing mammalian cells only in 2013. Jennifer Doudna and Emmanuel Charpentier were awarded the Nobel Prize just in December 2020 for their discovery of CRISPR and their adaptation of the system for gene editing purposes. In our application, we've taken the same AAV8 virus we used in the gene addition experiments, but we swapped out the PAH gene and we put in the gene for Cas9. Cas9 is actually a protein that's native to many bacteria. For bacteria, it's part of their defense mechanism against foreign invaders, namely phage viruses that like to infect bacteria. And Cas9's function is to locate and destroy the phage DNA. It's very discriminatory. It seeks out a very specific DNA sequence and cuts the DNA at that sequence. The beauty is that we can engineer the system to cut in a very specific site close to the mutation in the PAH gene and cut only at that site. Cas9 will frequently cut both chromosomes, but I'm just showing it cutting one. Now Cas9 just makes a cut and leaves a hole. Somehow a lot of people, even some scientists, have misunderstood that CRISPR-Cas9 editing can directly cure a disease like PAH deficiency, but that really isn't true. Cas9 just makes a cut in the DNA, but it doesn't fix the PAH gene. The cell itself has mechanisms that fix the hole, and we've learned that hepatocytes preferentially just stick the two ends of the hole together to patch things up. Well, that doesn't cure PKU. In fact, it makes things worse because now you've just basically wiped out a big chunk of the PAH gene. This technique is actually useful for making new disease models, and it's been used to make a PKU pig, and Dalen recently used it to make a new PKU mouse model. Anyway, to get back on track here, how do we get a fix instead of a bigger mess? Well, we can infect the cell with a second AAV, but this one carries a piece of the normal PAH gene without any mutations that matches the area that was cut. Now we've left out any promoter signals, and furthermore, it's not the whole PAH gene, it's just a fragment. We add this fragment, and our hope is that in the course of repairing the hole, the cell will incorporate this piece of the PAH gene. If this is done correctly, the patient's chromosome now has an intact full-length PAH gene without any mutation that encodes the correct sequence for PAH protein. So Dalen designed a CRISPR guide RNA that directed Cas9 to create a double-strand break near the PAH mutation in the PKU mouse, and she supplied a second AAV with a repair template. As I mentioned, the preference of hepatocytes is to use non-homologous end joining, or NHJ, to repair the double-strand break, and to just ignore the repair template that Dalen supplied to the mice. She used a trick to facilitate homology-directed repair, or HDR. She treated the mice with vanillin, a potent NHJ inhibitor, which forced some of the hepatocytes to use HDR instead of NHJ, and to incorporate the repair template into the damaged chromosome to repair the double-strand break. We also learned early on that unlike gene addition, this only worked if you applied it to neonatal mice. If you wait until they're adults, we only saw deleted PAH genes and no corrected genes. So Dalen injected infant PKU mice with a mixture of both AAV vectors with or without vanillin. She actually redosed them as adolescents, but then waited until they were adults to analyze the results. The immediate good news was that some of the mice as adults had turned black, looking much like their unaffected litter mates. These are the serum Phe results in 4 mice that had received both AAV vectors with vanillin, which are depicted in yellow. Untreated PKU and wild-type mice are depicted in blue and green, respectively. At euthanasia, when the animals were 18 weeks old, in the mice that had received both AAV vectors and vanillin, an average of 13% of the PAH alleles had been corrected with HDR, while the mean correction rates were lower than 5% in mice that received both AAV vectors, but no vanillin or had received only the repair template plus vanillin but no Cas9. In the fully edited animals, liver enzyme activity had been restored on average to about 10% of wild-type activity, and their mean serum Phe had decreased to about 600 micromolar. So we'd not quite achieved a complete cure, but we have some new approaches to try it, which we hope will further improve their repair frequency and lower the blood Phe levels even further. There are currently several ongoing clinical trials utilizing CRISPR-Cas9. The first 3 use the technology in bone marrow stem cells or circulating T lymphocytes, which are removed from the body and then treated. A trial was recently initiated at OHSU to treat a form of congenital blindness by injecting CRISPR machinery directly into the retina. But just this month, the first patient to receive a systemic injection of CRISPR reagents was treated in a trial for transthyretin amyloidosis. In this study, CRISPR-Cas9 reagents are delivered to the liver, but the goal is only to cause a deletion in the dominantly acting pathogenic gene. There's no need for HDR with a normal repair template. Repair in liver has yet to be attempted in a human. This is our overarching goal: to develop a safe, effective gene therapy method that will permanently produce PAH enzyme in a majority of liver cells, leading to permanent reduction of blood phenylalanine, even if the individual with PKU is consuming normal amounts of dietary protein. I do not do this work alone, and I want to acknowledge the small army of scientists at OHSU and at collaborating institutions around the world who really do the experiments. And of course, we do nothing without funding. I'm grateful for the core funding of the lab that we receive from the National Institutes of Health, and much of the work I've described is funded through philanthropic support from the National PKU Alliance. It's our hope that one of these novel treatments will someday significantly improve the lives of individuals and families who are affected by PAH deficiency. And I thank you for listening today.